All right, let's jump right into this explainer. Today, we're unpacking what is literally a double-edged sword, genetic engineering. We're looking at a technology that fundamentally alters the very code of life itself. Now, this has the incredible power to cure devastating diseases and feed billions of people, but it also carries the potential for some unprecedented ecological and biological disruption. So get ready, because this is going to be a deeply fascinating journey. Here is how we're going to break this down to truly grasp the topic. First, decoding genetic engineering. Second, the chemistry of recombinant insulin. Third, transforming our global agriculture. Fourth, the advantages of genetic engineering. And finally, number 5, its limitations, risks, and disadvantages. So starting with section 1, decoding genetic engineering and the basics of modifying life. For centuries, humans relied on conventional breeding, right? You cross related species, wait for natural mutations, and basically just hope for the best over multiple generations. But genetic engineering completely shatters those boundaries. If a favorable trait doesn't exist naturally, scientists can literally cut a specific gene from a completely unrelated organism and paste it into their target using recombinant DNA technology. We aren't just selecting traits anymore, we're intentionally transferring them across the entire tree of life. Moving right along to section 2, the chemistry of recombinant insulin. This is really a micro-level miracle. Because prior to the 1980s, diabetics were treated with insulin that literally had to be harvested and purified from the pancreases of slaughtered pigs and cows. To put into perspective just why that old method is entirely obsolete today, consider the massive scale we're dealing with. By the year 2025, it's estimated that roughly 300 million diabetics worldwide will require over 16,000 kilograms of insulin every single year. Now, if we tried to extract that much insulin from animals today, the sheer amount of land, water, animal feed, and slaughterhouses required would be logistically impossible. The carbon footprint alone would just be catastrophic. So how does the chemistry actually work to solve this? We map out a precise chemical procedure to turn E. coli bacteria into microscopic living factories. First, the human insulin protein's 2 subunits, chain A and chain B, are cloned into separate E. coli populations. We then insert them into the beta-galactosidase gene of a plasmid vector, so the bacteria produces this fused chimeric protein. Now here is where the pinpoint chemistry happens. Scientists use a highly specific chemical called cyanogen bromide to cleave or cut the peptide chain. This frees our desired insulin tails. Finally, we join these purified chains together through oxidation to create perfectly active, synthetic human insulin. It is an absolute masterclass in chemical engineering. Okay, section 3, transforming our global agriculture. Taking this from the lab straight to the field, let's scale things up and see how this exact same recombinant DNA technology completely revolutionized farming. It's honestly fascinating how a single bacterium, discovered way back in 1901 by a Japanese biologist who noticed it was killing silkworms, eventually transformed our modern fruit supply. That bacterium is Bacillus thuringiensis, or BT. Eventually, scientists realized they could take the gene that produces this toxic effect and transfer it directly into plants. By 1983, the first genetically modified tobacco plant was tested in a lab. And then in 1994, history was made with the FLAVR savor tomato. Now, this was engineered specifically for a longer shelf life, even if, admittedly, it didn't quite taste like the future we had hoped for. Let's look at some real-world agricultural data from India regarding BT cotton, which is engineered to resist pests. Now, while there are absolutely ongoing debates about the long-term impacts, today the historical data is pretty striking. Between its introduction in 2002 and the year 2020, BT cotton yields in India more than doubled, jumping from 191 kilograms per hectare to 436 kilograms per hectare. Which brings us to section 4, the advantages of genetic engineering, or solutions for a growing world. When applied effectively, genetic engineering acts as a vital tool to fight some of humanity's greatest existential threats. The pros here are massive, and they directly impact your daily life. Over in agriculture, we're engineering climate resilience against severe droughts and extreme heat, which allows us to grow way more food on far less land, all while slashing the need for chemical pesticides. This translates directly to a lower carbon footprint and reduced land and water use.
And in human health, well, beyond just insulin, recombinant DNA allows us to produce targeted therapeutics. Take Herceptin, for example. It's a life-saving, engineered antibody designed specifically to attack the genetic roots of HER2-positive breast cancer.
But we really need to impartially examine the other side of things. Section 5. Limitations, risks, and disadvantages. The complications of playing creator. Because, playing creator definitely has consequences. First up, there's gene escape, which is where modified traits accidentally crossbreed into wild weeds, creating these sort of super weeds. Then you have unintended allergies. A classic example is when soybeans were enhanced with Brazil nut proteins to improve animal feed, but they inadvertently carried deadly allergens for humans. Plus, nature adapts. Pests are accelerating their resistance. We're already seeing bullworms evolving to survive the toxic proteins in Bt crops. On a socioeconomic level, the corporate monopolization of seed patents creates immense financial hardship for smallholder farmers. And, of course, there is always the ever-present potential for unforeseen ecological disruption and harm to local wildlife. So those are just the accidental risks and the socioeconomic debates. But we also have to ask a much darker question. What actually happens when these incredibly precise chemical and biological procedures are intentionally misused? The CDC maintains very strict categories for bioterrorism, and Category C is particularly chilling in this context. It specifically defines emerging pathogens that could be engineered for mass dissemination. The frightening reality here is that recombinant DNA technology can absolutely be weaponized. Hostile actors could theoretically manipulate genes to increase a pathogen's survivability, its infectivity, and its drug resistance. That means creating a new breed of highly lethal biological weapons designed for mass spread and high morbidity, meaning severe illness rates specifically for modern warfare. In fact, historical memoirs have already detailed past programs that attempted to weaponize horrific viruses like Marburg. So we've mapped out the chemistry, we've seen the incredible data on crop yields, and we've acknowledged the very real ecological and security threats. Genetic engineering isn't inherently good or evil. It's merely a tool of immense, unparalleled power. So the ultimate question we face in the 21st century is this: how do we balance the unparalleled power to cure humanity with the catastrophic potential to destroy it? The code of life has been unlocked, but it is entirely up to us how we choose to rewrite it.